Good morning and welcome to my lecture. I'm Dr. Sonia Iverson and I'm going to be talking to you today about opportunities and experiences in biotech startups. Like many of you, I'm sure, I grew up in Montana in a very rural community called Sunburst near the Canadian border. My graduating class was 23 students in a town of around 400 people. We had 3 churches, 3 bars, one of which used to be a church, and not a whole lot else. But what we did have was Mr. Larry Fouke. And Mr. Larry Fouke had a program he called the Independent Science Investigations. Mr. Fouke was our science teacher, by which I mean he taught pretty much every aspect of science that we had in our small school. He was my chemistry teacher, my biology teacher. He knew just enough about everything to point you in the right direction so you could start exploring your own interests. His program, the Independent Science Investigations, or ISI, was a special class that was invitation only, where he handpicked students who had demonstrated a scientific curiosity to be in this program, where he trained us how to think critically about research, how to be curious about biology and chemistry and other aspects of research, and to chase those curiosities in a rigorous scientific manner. And as part of that, he led us in presenting our research at the regional and state science fairs, as well as some other symposiums. And it was a fantastic opportunity. It helped me not only to develop as a scientist, but to get to experience some traveling for my first couple of times away from home, and also to have to develop an idea on my own, to find something in research that I found interesting and pursue that, and learn how to seek those answers, and then be able to discuss them. It was formative, and I wish more schools had it. Through his program, I ended up competing with my research at the regional and state science fairs and earned my way to the International Science and Engineering Fair as both a junior and a senior. And this international fair hosts a couple of thousand students from around the world, judged by scientists from around the world in all different fields, everything from environmental biology, chemistry, macrobiology, microbiology, you name it, physics, all of it. And I had the wonderful opportunity to compete at this fair twice. I could say a lot about ISEF, but probably the main point is that this was my first time really standing in front of my research and explaining it to somebody else, was through these science fairs, and learning not only how to do research, but to communicate it as well. From high school, I went to Bozeman. I'm sure for many of you, there's many reasons to do so besides the beautiful mountain views, as well as potentially proximity to family, and that's kind of what drew me there as well. But part of it, what drew me to Bozeman, was that I knew they had a strong science program, particularly in biology research, and that's kind of where my interest was, but I wasn't really sure. At the time, I was actually studying psychology because I thought I was going to be a social worker. So in the end, I got my bachelor's degree in biotech animal systems, in part due to Dr. David Sands, drawing me back into biology when I thought I was heading down a different path. And I also was lucky enough to finish my minor in genetics the first semester that it was offered at MSU as well as a minor in psychology. When I finished school in Bozeman, I stayed in Bozeman for a few years working as a researcher. I'll talk about that a bit in a moment, before going to graduate school. But when I decided to go to graduate school, I wanted to get a bit of a change of venue. I wanted to experience life in a different setting. At that point, I had grown up and lived my whole life in Montana, first in a very rural area and then in Bozeman. And so by the time I graduated and was ready to eventually go to grad school, I started looking at universities on either coasts, and I ended up in Boston. So Boston is quite a bit of a different perspective, a different venue, if you will, and especially in the sense that the campus, Bozeman, you guys have this wonderful campus with the trees and the mountains in the background. The campus, it's a little bit covered by my picture here, but you'll get the idea. This highway, or major road right here that you can see in the foreground of the picture, that is Boston University's campus. It runs along a major stretch of road. So it's a bit of a different atmosphere. However, it was a very interesting learning experience, and it eventually brought me back to Boston some years later, so I guess it must have worked out well. I earned my PhD in cell and molecular biology and biochemistry, as well as my master's, and I did that research in essentially a synthetic biology lab. And synthetic biology is an interesting field. I'll discuss some more in the following slides. But mostly, I'll leave it to say that it's a combination of computers and biology and engineering. So before I went off to graduate school, actually while I was an undergrad, I was looking for work in a research lab. And Ed to this day will tease me because when I interviewed with him to join his lab as an undergraduate research assistant, I told him that I only wanted to work with him for 6 months because I was convinced at the time that the best way to become a well-rounded scientist was to be a bit of a jack of all trades, to learn a little of this and a little of that so that you could make the giant leaps between two fields. You can use one field to influence your thoughts on another. And I still believe that these kind of cross-disciplinary approaches are going to give some of the best outcomes in research. However, as a potential boss, it was not an inspiring thing to hear that your potential new employee only wanted to work for you for 6 months. Like, what's the point in training somebody at that point? Well, luckily for both of us, I never got bored. I worked for Ed from 2004 until I went to graduate school in 2011. And every month, every year was a different experience, doing different research in different fields, learning different techniques. And if any of you are interested in getting into research at MSU, I would highly suggest finding this guy and seeing if he's hiring. He's a fantastic advisor, and I learned a lot from him. One of the things I learned from him is that titles of papers often don't tell you a whole lot about what's in the paper, or they do, but in a roundabout sort of way. Personally, I like to call this paper the reminder that the liver regenerates. Unfortunately, that wasn't a good enough paper or title for a scientific journal, so instead, we just got pretty red and green pictures that made for great Christmas cards. However, the cooler story, the deeper story here, is that we were able to use green fluorescent proteins and red fluorescent proteins expressed by mouse liver to determine a lot of things, but namely to determine how liver regenerates. There, at the time, was an assumption within the field that once the liver hits an adult stage, all of the development of new liver cells came from old liver cells dividing and just making new liver cells. And that is true to a large extent. The liver is one of the only tissues in the human body that does pretty much endlessly replicate. Most other cell types in the body get to an endpoint where they stop replicating and they stop replicating and stop differentiating. Hepatocytes, on the other hand, will continuously replicate and make new hepatocytes. Hepatocytes being the main form of the liver cells. What we were suspecting, however, was that there had to be a basal stem cell contribution to new liver cell development, new hepatocyte development. And in order to identify that, we created these red-green stoplight mice. And so what happened here with these mice was that in every other cell in the mouse, these cells were red. And the construct for this is basically we engineered a piece of DNA that was put into the mice where it always expresses a red fluorescent protein unless that little piece of DNA gets cut and modified, and then it produces a green fluorescent protein. It only gets modified in the presence of a certain protein called albumin. And albumin is only expressed by hepatocytes. So in mice, we could actually measure the change from red to green to determine when a hepatocyte was newly born from a non-hepatocyte precursor. And so in these pictures, what you're seeing outlined in red is the membranes of all the hepatocytes in a piece of mouse tissue. And here we artificially gave these animals albumin and essentially triggered the color change. And just so that we could see how long after we triggered that color change would it take for the cells to turn green. And then once we had this, we call it a chronometer, once we had this measurement of time of how long it took for the cells to turn green, we went back and looked at actual mouse tissue. Well, this is also actual mouse tissue, but mouse tissue that we haven't been modifying in that way. And what you can see here, I hope you can see in these pictures, is that in a normal mouse, in B and C, most of the cells are green outlined with only a couple of red outlined cells right here. And then and that's in a young mouse that's still developing. And then in an older mouse, you can see from this picture, there's fewer of these red ones. So the yellow is pointing at all of the red cells, the yellow arrows. In an adult mouse, there's fewer red cells. However, if you take an adult mouse and you do what's called a partial appendectomy, which is the same thing that would happen if you had like a liver tumor, they can cut out part of the liver tumor and your liver will actually grow back to replace that lost mass. And this is something you can do in mice to essentially mimic liver damage and regeneration. And so in a mouse, if you cut a piece of the liver and then allow the liver to grow back, in that regenerating liver, it looks more like a juvenile liver with a lot more red cells. And so these red cells then were indicative of hepatocytes that had recently been differentiated from a non-hepatocyte precursor, a stem cell, essentially. And this was really cool because at the time it went against the dogma in the field. And even now, this is something that it's understood and accepted by some people in the field, but it's still a little bit cutting edge, a little bit new. One of the other projects that I worked on with Ed that I thought was amazing at the time, it's one of the clearest cut pieces of data I've still to this day ever seen in research, was a paper we published called the thyroidoxin reductase 1 dependent metabolic switch alters hepatic lipogenesis, glycogen storage, and detoxification. Which, remember when I said titles don't always tell you what you want them to? I mean, the title tells you a lot, but I think the take-home message was much cleaner, which was superpowered mutant mice survive Tylenol overdose. So in this mouse model, for other reasons, we had made a knockout mouse, meaning these mice did not express thyroidoxin reductase 1. And this was done for a completely different project looking at oxidative stress. And as a side note, at one point in this project, we had noticed that these mice behaved a little bit differently when they were challenged with a drug. When we were using a drug in a different experiment, we had noticed that these mice behaved a bit differently. And so we started to consider that maybe they handled drug challenges differently in general. And what you can see in the left side here where it says control no APAP, APAP is the abbreviation for Tylenol, and the picture on the far left, the C and then A, is a wild type mouse under normal conditions, a heterozygous mouse, meaning it has one knocked out gene and one wild type gene under normal conditions, and the G panel here is a knockout mouse, so it's null for thyroidoxin reductase 1 under normal conditions. And all of these pictures look fairly similar. This is all healthy liver tissue. However, 8 hours after challenging these mice with a fairly strong dose of Tylenol, a dose equivalent to what would cause liver damage in humans, you can see all these red patches, these pinkish red patches in the wild type mouse, which is indicative of necrotic and apoptotic cells. So this is cell death happening in the liver. And this is what happens in humans with a Tylenol overdose. In a heterozygous mouse, you do still see some of that pink, but it's not as bad. There is some protection. And in a knockout mouse, if you compare this G image to this G image, it's almost identical. And what we took away from that was that our knockout mice were almost entirely refractory to a Tylenol overdose, meaning that they somehow managed to not be damaged by a Tylenol overdose. And when we looked at the gene expression in these cells, in the wild type versus the mutant cells, without a Tylenol challenge on the left, you can already see that the null mice have a very different gene expression pattern. So if you're not familiar with these kind of tables, basically each of these little bars represents a single gene that we're measuring the expression, the activity of that single gene. And 1, 2, and 3 are wild type mice, and 4, 5, and 6 were 3 different mutant mice. So we can get some redundancy in our measurements. And what you can see is that the blue represents a decrease in gene expression compared to the wild type, and all the yellow bars represent an increase in gene expression compared to the wild type. So there's a lot of different gene expression happening between mutants and wild types. And then when we treat them with Tylenol with a lower dose, because we treat it with too high of a dose, then the tissue sample doesn't work as well for this assay. But when we treat them with a lower dose of Tylenol, we could see some different changes in the gene expression pattern. And when we dug deeper into that, what we were able to find out was that our thyroidoxin reductase null mice had a significant metabolic shift. Their gene expression had changed to adjust the way that they metabolized glucose and a lot of other things. It had also turned up the drug metabolism pathways. So the reason why these mice were refractory to Tylenol was because the thyroidoxin reductase null mice were much more efficient at exporting drugs from the cells. And so the Tylenol had less time to become damaging to the cells. We thought, going into this experiment, we were really hopeful this was going to lead to a treatment for Tylenol overdose. Unfortunately, it turned out that the effect of the knockout was a long-term systemic effect. And so there's a drug on the market that mimics this knockout, but it doesn't work in the same fashion when it's given as an acute treatment. It would need to be a sustained treatment. And you can't generally treat someone for a Tylenol overdose before they have an overdose. So unfortunately, this didn't turn out to be a treatment, but it did give us a lot of insight on how Tylenol overdoses work with relation to these different pathways, but also how thyroidoxin reductase has a role to play in drug metabolism and metabolism in general. So eventually I graduated MSU after publishing a few more papers with Ed, and I stayed and worked as a research associate and then a lab manager in his lab for a few more years. However, eventually I got the traveling itch. Like I said before, I had grown up in Montana, spent my whole life there, but I got a touch of travel in high school and a little bit more in college, and eventually decided I wanted to set out on a bit of an adventure. And so I did, while I was working for Ed, I took some time off and I did my first trip abroad, traveling to Syria through Europe, but through Turkey and Syria. And I worked actually in Syria at the International Center for Agricultural Research in Dryland Areas, ICARDA, which is a nonprofit international agricultural research organization that Dr. David Sands had set me up with as a visiting scientist. And this gave me an opportunity not only to see a bit of the Middle East, but to explore the possibility of doing research in that field and in that region. I eventually did decide to come back to the US. I went back to work with Ed for another year while I considered graduate school. But at this point, I definitely had an itch to travel, a wanderlust, if you want to call it that. So a lot of my decisions from here on out had a lot to do with travel. And one of the first things that came up was the toothpick project. So while I was still working for Ed and I was applying for graduate schools, I asked David Sands, Dr. Sands, if he could advise me on my graduate school applications. And when we met to talk about graduate school, he kept talking about Kenya. And next thing I know, we're buying a flight for me to travel with him to Kenya and help to organize a research project there in his efforts to show the efficacy of a treatment for a corn pathogen. And I'm sure he's talked to you guys already about Striga and the toothpick project, but just in case he hasn't, the basic story here is that throughout Africa there is a obligate plant parasite called Striga that kills the corn plant or steals nutrients from the corn plants. And has a devastating effect on the yield of corn harvests across Africa. And corn is one of the major food products, one of the major agricultural products, and kind of a staple of basic life for a lot of these farmers. So the toothpick project was based on some research that Dave Sands had done, where he identified a natural fungus called Fusarium oxysporum, he calls it Foxy, which was able to inhibit the growth of Striga and allow the corn to germinate without being essentially waylaid by the Striga. And so the reason why I was traveling with him to Kenya was to help organize a research project and help teach the local women that he was working with the ideas of scientific research and scientific principles and how to structure a research project in order to get scientifically valid data. And that's what we did. We spent about a week in Kenya, at Masino, Kenya, teaching these women the basics of the scientific principles, scientific method, and helping to organize the research, the plots to study the different variants of Foxy that he had brought down. These are just a few more photos from that trip. This is still my only experience in Africa, and one of my first and favorite trips abroad. We got to visit a bunch of different farms. We helped these ladies planting the fields. This is the family that we stayed with. This photo of the kids always makes me smile because there wasn't a whole lot of communication ability between them because I didn't have really any Swahili. They didn't speak a lot of English, but they understand cheese. And so I was trying to take pictures. They always smile, but I kept having a hard time getting the kids to look at the camera. And so finally they're being shy. They're looking away. Finally I said, look up. And I nodded my head like that. I said, look up. And this is the photo I got. Two of the kids took me at my word and looked straight up. And the third kid decided to be a little silly and tease me by looking straight down. It just goes to show that you don't necessarily need language to communicate and have a good time while you're traveling. And that was probably one of the best lessons that I took out of my trip to Kenya. All right, now I'm going to jump ahead just a little bit because shortly after I went to Kenya with Dave, I joined the biomedical engineering program at Boston University to do my PhD. And I was working in the CIDR lab, the Cross-Interdisciplinary Design Automation Research Lab, run by Doug Densmore, my PI. Doug is a computer scientist who was interested in using computers to help aid biology research. And in that effort, he had built out a lab at BU that was mostly computer scientists, and they were just starting to experiment with some biology. And one of the things that they were working on was designing software to help biologists who are building DNA, essentially, putting together pieces of DNA to do interesting things. So like with the mice work that I was doing before, we had to construct DNA that would express these red and green fluorescent proteins and put those into mice. And the means by which we did that was very old school and very engineering heavy in a way. Going forward, as biology has progressed, we've developed some really cool tools around how to engineer DNA in the lab, but there's not a lot of computational tools, especially back in when I joined this lab, there wasn't a lot of computational tools to help design DNA designs. So one of the things that I was working on as one of the first students in his lab, sorry about the animation there, I was one of the first biology students in his lab, was essentially building the bridge between biology and the computational side. And so his group, the computational side, was trying to design software that would help scientists design their DNA constructs, but they didn't know a whole lot about DNA and working actually in a wet lab, working with DNA and plasmids and bacteria and cells and all of that. So I came in and tried to help kind of translate the computational biology, into wet lab biology and back. So just for a quick bit of terminology, and also to show you how we tend to look at these if you haven't seen this kind of stuff before, when we talk about DNA, we often draw it in lines and shapes like this because it's much more complicated to look at the actual chemical structure. And so oftentimes in a paper, you'll see something like this, a square, like a rectangle representing a piece of DNA and then annotated like this. So, and then when that's in a plasmid is a circular piece of DNA that is grown in bacteria. And this is how bacteria essentially pass DNA between different cells and then replicate their own genomes. The way that we tend to annotate in synthetic biology is to label a promoter as this little arrow. A half circle is a ribosome binding site, so the promoter tells the cells where transcriptions should start. The ribosome binding site tells the cells where translation should start, so where to actually make the protein off the mRNA. And then the coding sequence is the actual DNA sequence that is converted to an RNA sequence that is then converted to protein. So it's that portion of the sequence that gives you the actual amino acid sequence. And then the terminator tells it all when to stop. And when we put that into plasmid in synthetic biology, we tend to make it look like this. Now, for the sake of what I'm going to show you here, we actually refer to this even a little more technically because they have to have fancy names for things. And we call this a transcriptional unit, meaning that this is what you need, a basic transcriptional unit. This is a minimum amount of DNA that will tell a cell how to express a certain protein. All right. So this is something that Doug was working on at the time, and his computational team was this user interface and the logic behind it, where you could tell the computer that you wanted a piece of DNA that would express a green fluorescent protein only when it detected this particular sugar molecule under a pH that was this high or low, for example. and basically trying to treat biology and to treat cells like machines that could be programmed, like computers that could be programmed. And this eventually got published in, I believe it was Nature. It was either Nature or Science, but I think it was in Nature around the time that I graduated. The software is an existing tool now, and it's being used by some companies as well to help design their research. But at the time, they didn't actually have anybody trying to build these structures in the wet lab and test whether or not they actually worked. And so that's kind of where I came in initially, was just trying to help set up the wet lab and test whether or not these programs worked. Apologies, the slide got a little bit messed up, but you can mostly see it. This shows basically a promoter in a plasmid, a ribosomal binding site in a plasmid, etc. And then all of these could be combined into one plasmid that has all four components. And the reason this is important is that we're basically taking something that in biology, in regular molecular biology, is not seen as these very defined and structured parts. And we're making them into defined structured parts and then being able to assemble them in a fashion where I can take all four of these at once in one tube and cut these pieces of DNA and then treat them with temperature and some enzymes and end up with the final product that has all these pieces of DNA in the right order in the final plasmid. And so this is what I was developing in my graduate program in order to enable the kind of software automation that the computational side of the group was doing. And then there's a second level to this. You can take each of these transcriptional units then and combine them to make a larger circuit. All right. So this was the overall concept of the base level of my PhD, was developing this system, creating a bunch of parts, so taking parts that kind of existed elsewhere, adapting them to the system, testing them, developing a few new parts, and providing all of this as a kit in like a 96 well plate that can be provided to a lab as a starting point to build these sort of systems. So this is a large portion of what I did in my PhD. And so the reason any of this is even interesting is because if you're designing things like what we were doing in mice before when I was working for Ed, that's generally kind of like a bespoke design. You're making something really custom and you're making only one of it or a couple versions of it, but each one are very different. So you kind of have to do each one by hand, individually, in this like bespoke manner. However, there's a lot of reasons why you might actually just want to be tinkering with one design and changing one piece of it. For example, there are some drugs, some molecules that we know that are medicinal that are made by plants or microbes, etc. And maybe they're not made very efficiently, but we know that they're made by another microbe. And so what we can do is we can actually take the genes that make that drug and we can tinker with them and we can try and optimize the expression and to make it more effective to produce a drug that way. And there's a few different companies that have been doing this for various, like anti-malarial drugs is one example that's been being produced in yeast like this. And anyways, if you're doing something like that, you need to be able to balance the expression of multiple genes, generally speaking. Another example of this kind of thing would be like golden rice, for example, where the rice is, it takes 4 or 5 different genes to produce beta carotene in rice, and they have to express at certain levels. And so if you come in and you play with the promoters or the ribosomal binding sites, which control the amount and the timing of gene expression, you can kind of fine-tune the control of that construct. And so one of the ways to do this would be to manually build a bunch of different versions of the same construct with just a little bit of a change, and do each one of these as a separate thing. But through a system like this MoClo assembly system that I've been discussing, you can actually do all of this at once. And so because only the, I haven't really discussed the annotation here, but only the orange dots can connect to the orange dots. And same for like green dots to green dots, etc. So you can actually take 5 promoters at once and cut these pieces out of their plasmids, and it'll only connect in the same order. So you'll end up with 5 different plasmids, but they won't rearrange in the wrong pattern. And because of this, you can actually make a lot of constructs at once. So you can create a library of DNA variants in a single tube. And this can be really helpful for characterizing different proteins and how they work, for screening a mutant library where you want to try and change how a coding sequence works to see if you break this amino acid, if you change this amino acid, what does it do to the protein? You can build that whole library at once in one tube. Or like I said, for tuning the expression levels. And this can get a little crazy. You can make some very large libraries through this combinatorial process. So with the original Moclo, building a library like this, one piece at a time, with the original protocols, would have cost over $2,000 and would have taken 16 hours of thermocycler time and 3 hours of pipetting time. With the modified systems, where the protocols I adapted to improve them, now we can do this in 4.5 hours of time in the thermocycler and about 3 hours still of pipetting time. But if you can do this all multiplex, if you can set this up so you can do it all in one reaction, then it takes only about $1.50 in reagent costs in 1.5 hours in the thermocycler and about an hour of hand time to produce all of these different constructs. So that's the powerful aspect of something like this combinatorial design with a multiplex assembly. And obviously, these are much faster and cheaper when you can do it this way. All right, so that's a very quick overview of what I did in my PhD. I used that system then on a couple of other projects, including helping a biology group that did mouse research where they were studying aldolase, but they were trying to express 2 different versions of aldolase at the same level and having some difficulties. I was able to use the data I had produced and the system I had produced with the Moclo to help them fine-tune the expression of those 2 proteins and be able to move forward with their research that had been stalled previously. So there was some real applications I was able to do in grad school with that program. But by the time I graduated, I was ready for a change of pace. I kind of missed the more hard science research I had been doing with ed, but at the same time I wasn't quite sure what I wanted to do next. And so for the next about 3 years, I actually traveled. This map that I've shown a couple of times, each of these dots is a place that I have been, that I've traveled. And I did a lot of that travel over this 3-year period after graduate school. Of course, travel isn't free, and so while I was traveling, I had a variety of freelance consulting jobs. At first, I was working with a small chemical synthesis company providing technical scientific support, which meant basically that they had customers who wanted to produce a particular small molecule or had some biology issue that they were trying to work through. And I would call these scientists and talk to them and try and help them solve their problems. Another company that I worked for was a biochemical reagent company that was producing a particular type of ELISA or antibody assay, and I helped them produce technical marketing materials. So this is actually a little bit of graphic design in biology and science. But then I really kind of struck gold on a project that I very much enjoyed when I connected with a venture capital firm in California and was able to dip my toes in the investment capital world, essentially. And so through that, I was able to learn a lot about investments and how to investigate a startup company and evaluate whether or not that startup company was going to be viable, whether or not their research was good. And it's just a lot of different aspects of how venture capital and startups and biotech work. So the venture capital firm that I worked for was called Civilization Ventures. And it's a small firm that has recently just raised their second fund and has invested in a few dozen companies, but some of those companies have done really well. One of the companies they invested in was called Rocket Pharma, which is working on gene therapies and went public a couple of years ago, or a year and a half ago. A couple of other companies have been bought by larger DNA-related companies and are doing really well. But what I really enjoyed about working with Civilization Ventures, and I could do this while I was still traveling, was I had the chance to look behind the curtain on these startup companies, evaluate the research that they were using to start their companies. And not only did I get to keep abreast of really interesting novel science, but I got to kind of dig into the details of different areas of research that I might not ever have been drawn to on my own. One of the investment memos that I helped with was on a machine learning company, for example. I helped evaluate a potential investment into a company that wanted to use machine learning to study, or to evaluate small molecules that could potentially be used for therapeutics. Probably more in my field, I've gotten to also work on their investments for Singular Bio, which has been a successful investment for them. Singular Bio uses cell-free DNA testing to look for early stage cancer from a blood draw, which is pretty spectacular that they can do so with the kind of precision that they are using. And then Durham Biont is a company that I also helped with the investment memo on to evaluate whether or not they can make a treatment for foot fungus, of all things, using microbes. So they have a theory that the reason why toenail fungus is a persistent problem is because it's due to a disruption of the microbiome in your feet. And they're trying to develop therapeutics that are actually bacterial treatments. And I think microbiome therapeutics is something that is really up and coming right now as well, and something I have a little bit of familiarity with, mostly through some friends who work in the field. And I think that we're going to start seeing some really interesting things there in the next few years. So one of the cool things about Civilization Ventures recently is that they just finished raising their second fund. So I don't know if you guys know anything about venture capital, but basically how this works is that they recruit funding from a variety of sources and make one pooled fund of a large amount of money that then the venture capital firm is responsible for choosing the investments. And a little bit like a hedge fund, I guess. And so the second fund was just raised at a tune of $35 million and is currently being invested in small startup companies. So around that, after about 3 years of traveling and working as a consultant, I started dipping my toes back into the idea of doing research again. And eventually I ended up getting a job as a scientist with a company called Sana Biotechnology here in Boston, which is where I'm currently working. And Sana is a combination of gene therapy and cell therapy. And I've been working here for about 2 years. I recently got promoted to senior scientist, and I've worked on a variety of projects related to making our gene therapies safer and stronger and more efficient, to treat specific diseases in either hepatocytes, so liver biology, or I'm just starting to get involved in some work on some cancer projects. So one of the things that we're working on at Sana that I find really interesting is in vivo gene therapy. Some of the earliest gene therapies that were successful were actually done 25-ish years ago, maybe 30 years ago even. It's been quite a while since we did the initial gene therapies on severe combined immunodeficiency, or the boy in the bubble syndrome, where these children actually had no immune system whatsoever at birth and often would die due to a common simple infection very young. And it was eventually discovered that that mutation was on the X chromosome. It was a single gene mutation, very easy to fix if we had the tools to fix it. And so I think it was about 25 years ago, they did what was called an ex vivo gene therapy, meaning not in life. And what ex vivo gene therapy is where they actually take cells out of the body and use a virus to change the DNA, and then you select the cells that have been changed and put them back in the body. So the virus itself doesn't go into the body. And that worked for these kids that were dealing with severe combined immunodeficiency, the boy in the bubble syndrome. And it basically took kids who had no immune system at birth and would have died of a basic infection and gave them a fully functioning immune system within a few weeks. However, 2 or 3 of the kids over the course of a few years developed leukemia. Granted, these kids wouldn't have lived to be a few years old without the treatment, but it still wasn't an ideal outcome. And what they realized was that the mechanism, the type of virus they were using, had a tendency of taking the DNA that it was supposed to deliver and putting it in particularly bad locations in the genome. And so in those cases, the DNA that was being added to the cells to help treat the disease was sometimes landing in a cancer gene and causing cancer. The cancer was generally treatable and much more treatable than the boy in the bubble syndrome, but it caused some concern at the time around the idea of gene therapy and safety of gene therapy. And so that was happening not too long before I went to college, that I was kind of in the impression that gene therapy was going to have a long, hard road to becoming acceptable, essentially, to becoming safe enough. And what's really cool now is that working at Sana, this is what kind of got me into working at this company, was this idea that we could now do in vivo gene therapies. And basically, over the years, we've learned more about how viruses work, how gene therapies work, to the point that we think we can now actually add the viruses directly into a human body and have them only fix what they're supposed to fix and not cause any problems. And of course, that's tricky, but I think that's where we're at. And so the idea here is that you have these very simple systems where you take a packaging vector, an envelope vector, and a transfer vector. You don't need to worry too much about that, but basically you take the pieces of a virus in DNA, the DNA pieces that will make a virus, but they only make a virus that can cure the disease, but not a virus that can continue to replicate and be pathogenic. And it won't spread from individual to individual. It's a hampered virus. And you put those into cells in the lab. And you make basically the cells will make a bunch of viruses for you. And then you can collect those viruses and actually use them as a treatment by injecting them into the patient. And this is one of the things we're working on at Sana that I'm really excited about. It's a kind of a new field that's up and coming, really big right now. So like in vivo gene therapy is one of the most popular things in biotech at the moment. And if we can make this work, it can be life-changing for a lot of people. Gene therapy in general has a lot of potential. It can cure rare diseases because a lot of diseases have a known genetic cause that could be a simple fix. But previous to gene therapies being a functional way to fix them, the only way to treat a lot of these diseases would be through treating the symptoms, so developing some kind of a drug that can treat the symptoms. And drug development is incredibly expensive and was really only financially feasible for diseases that affect many people because the drug company has to make back the money that they spend on developing the therapeutic. If we have the more that we develop our ability to do gene therapies and the more that those abilities become automated and streamlined, then we have the potential of being able to treat pretty much any disease almost in a bespoke manner. Because the dangerous part isn't necessarily treating, like actually adding the DNA in. The dangerous part isn't necessarily what the DNA is that we're adding in, it's how you get it in. And once you have the safe mechanism for getting the healthy DNA into the cells, it doesn't really matter what that DNA is. At that point, you can cure just about any disease that has a known genetic trait where the gene is broken, and all you need is a fixed version. And there's a lot of other techniques too that are being developed right now. I'm sure you guys have all heard of CRISPR, which can do a different kind of DNA repair. So CRISPR can actually change DNA at the site of damage, whereas the kind of gene therapy that I'm working on doesn't actually try to fix the broken DNA, it just adds the correct version of the DNA to the cells. One of the other things about the type of gene therapy that we're doing that's really interesting is CAR T, which some of you guys may be familiar with, which is the chimeric antigen receptor T-cell therapies. But CAR T therapies currently work as an ex vivo gene therapy, the way I was just describing for SCID, where they take cells out of the body of a cancer patient, genetically alter them, and then return them to the patient. The problem with this is in order to do that, it's a very difficult treatment for the patient. The patients have to be healthy enough to essentially survive the treatment because they have to take a lot of cells out, which makes the person weak, and then when you put the cells back, you have to kill off a lot of the cells that you're replacing in order to allow them these immune cells to survive. And so it's complicated and it's dangerous for the patients. So there's a large number of people who aren't able to do CAR T therapies because they're too sick. However, an in vivo CAR T therapy could be applied to a lot of patients who are too sick for our current CAR T therapies and would therefore save a lot more lives. All right, I'm going to try to kind of sum up. I know I've been bouncing around a little bit, but I wanted to give you guys a bit of an overview. Academia versus industry. So academia, some of the differences that you're going to find, there's pros and cons on both sides. In academia, you tend to get to ask your own questions and search for answers with some guidance from your advisors, or as you work your way up the ladder, you get to choose your own research in general. In industry, let me stay with academia for a minute. You also get to kind of choose your own adventure, which field you want to be in. There's somebody everywhere doing any kind of research. So if there's something that interests you, you can probably find a way to study it. You can have an exposure to a wide variety of research by working in an academic institution where the person across the hall is doing something completely different than you. You get to talk over coffee. You get a lot of exposure at conferences to different kinds of research. In academia, you get to focus a lot on the biology. The question is kind of the most important thing. It doesn't necessarily have to have an outcome other than biological curiosity or research curiosity. One of the downsides, of course, to academia is the pay tends to be lower, but you do often have a lot more freedom, both in terms of what your work schedule is like, whether or not you have time to travel, going to conferences, etc. And the other downside to academia, and one of the things that caused me to leave academia personally, was that it is not very well supported, unfortunately, at this time. And so grant writing and funding issues take up a lot of time and distract from the actual work and can be a lot of stress. However, there's some of my favorite times and my best memories in research were in academia. On the other hand, in industry, the focus is generally on solving problems. You might have less control over the research that you're doing in terms of the actual experiments because there's a goal to get to and a timeline. But you also tend to have a lot more resources to pursue those questions. You tend to sort of hyper-focus on a specific problem. So rather than learning to be a jack of all trades, you might learn to be a specialist. And for some people, that's great. And some people would prefer to have a wider range of skills. In industry, you often have to focus on the product. So the timeline matters, the product matters. That doesn't mean that the biology doesn't matter, because the biology is often the product. But you do have this focus on a goal that you don't have the same way in academia usually. In industry, you'll have better pay and better benefits, but the company and potentially your own research interests are beholden to the investors. All right, and then the last thing I wanted to bring up is part of the reason I wanted to talk to you guys today is because I feel like a lot of people who come from a small school, small university, a rural area often don't feel like the rest of the world is available. Maybe it's hard to see yourself in a different place. It's hard to see yourself playing with the big dogs, getting out and doing the most interesting things. And the other aspect is that maybe science isn't all of what it's about for you. When I came to MSU, I had every intention of being a social worker. I was studying psychology, but I missed doing the research that I had done in high school. And for much of my early adult life, I was struggling with this idea of being a scientist and. Whether that was a scientist and a social worker, or for a while it was photography and art, I couldn't settle comfortably on being just a scientist. And so what if that's not your only calling? What if you want to do more? And what if you're not sure if you can do more? And so that's where I wanted to say that basically you can do whatever you want. And I want to talk very briefly about another project I did especially over the few years after graduate school and while I was traveling. I've also been working with a nonprofit organization that I've created and a bunch of volunteers from around the world to teach sport to refugee kids. Oftentimes this felt rather silly. I earned my PhD to be a scientist and try and solve the big problems that way. And here I was literally playing in the dirt with kids. That felt sometimes like it was a bigger way of solving problems, a more meaningful way of solving problems. And this is a program that's still ongoing, although we've had to pause our activities for 2020 due to Corona. However, it's a fantastic way of connecting with people, of traveling, and of doing good in a place that a lot of people are afraid to even go. And so I just want to mention a little bit more about this. Why we teach slacklining, which is a balance sport, because it trains concentration and focus. It encourages students to attend classes. It teaches social skills like sharing, waiting for your turn in line, and teaches mindfulness and provides emotional control, because you can't balance very well if you're stressed out or dealing with emotional issues. We tend to work in Lebanon, although we have projects elsewhere as well, and the majority of our work has been in this white circle on the border of Syria. And the reason why we work here is because Lebanon has the largest proportion of refugees per capita of anywhere in the world due to the Syrian conflict. And so in a country of about 4 million Lebanese people, they had, at the peak, had about 2.5 million Syrian refugees. In theory, there's 1.5 million that are documented. And so there's a huge number of refugees here, a huge issue. And part of that issue is that they don't have enough schools. And so we were partnering with educational organizations and community groups to provide healthy recreation activities to support the school programs. And then lastly, I just want to leave you guys with this idea that you can be whoever you want to be. You can study whatever you want to study. You can do whatever kind of work or research or invention that you want to do. When there's no road, you can make your own path. And as a last bit of evidence, if you've seen this commercial, that's me. Any questions? Obviously, I can't hear any questions you might have right now, but feel free to reach out at my email address, just sonia.iverson@gmail.com. You're also welcome to reach out to me on LinkedIn if that's of interest, and I'm happy to answer any questions about the opportunities that exist in biotech, in industry, in academia, questions about grad school, for those of you who might be considering it. And I strongly suggest that if you're interested in research, you find one of the groups at MSU and see if you can join a lab, because it is a life-changing experience and it'll set you on a path that you could never guess. So thank you, and have a good day and stay safe.